Good day, students. Welcome to the lecture recording for Module 6, Reaction Energetics and Food Enzymes. My name is Miss Lusani Vangani, and this is aimed for the subject Food Chemistry 2. Before dwelling into the actual content of the work, just a reminder about the lesson plan that we actually follow throughout the entire chapter. What's important is the content that has to be covered, and then more specifically for you as the student are the learning outcomes. So whatever activities that we're going to do here in this chapter, make sure that it actually matches what you have to learn and it helps you to understand. More important are the references. Like I always mention, everything that is in this lecture has been summarized from certain different sources. So those sources are there right in front of you, and you actually have to go and consult those so that you can get in-depth understanding of the work and not just to rely on this PowerPoint presentation. Okay, so the chapter outline. This chapter is quite a big chapter, so it's divided into different chunks, like 3 chunks. So for now, the red highlighted areas will be what I'm going to be discussing in this recording. But all in all, what I'm going to be talking about throughout the whole chapter would be basic introduction of enzymes, and then how are they classified, and then from there we'll talk about the enzyme structure and then enzyme function. And then the next recording will focus on energy changes during enzyme activity, and then which then will boil down into the enzyme kinetics. And then lastly, I'll be talking about the factors affecting the reaction rate, as well as dwelling into how enzymes are actually used in the food industry, and talking about some different types of food products whereby enzymes are added or are used in order to aid the production or preservation of those food products. Okay, so in terms of the introduction, what are enzymes? Enzymes are proteins. So if we say they are proteins, it means they are made up of amino acids that are joined together to form a polypeptide chain. And that polypeptide chain undergoes the primary, secondary, tertiary, and quaternary structure. So it has what we call the native conformation. It's folded. And that is why we usually say most enzymes are globular proteins. So what do these globular proteins do? They are what we call biological catalysts. Of the catalysts that we know, we are used to chemical catalysts. I'll make an example of one in food analysis. When we are analyzing for the protein content of food, we usually use copper sulfate to speed up the breakdown of those proteins. But that is referred to as a chemical catalyst. The enzymes are biological catalysts because they speed up reactions in living organisms. Different living organisms. It can be human, it can be animals, it can be plant, it can be sea animals, anything that is living under the earth. So when they're doing that, they involve all the different chemical reactions whereby it's either they break down large molecules. Think about yourself and myself when we are eating food and the food has to be digested. Let's say you're eating starch, you just ate your bread. The starch is going to be broken down into simpler sugars, and those sugars are then going to be absorbed so that the sugar will give us energy later on. So that is known as the breakdown of molecules, and it's known as catabolism. In another instance, that's when you actually have those simple monomers like your proteins that have been broken down into amino acids. So those amino acids, when absorbed in the body, they are built up again to give us muscles. So that is called anabolism, and that is making up molecules. So in this entire process, the enzymes are involved in what we call the metabolism of all chemical reaction taking place. It is actually estimated that there's around about 6,000 different enzymes in nature. And those, we are actually talking about enzymes that have already been discovered. We don't know about new ones that people are still studying, that they will obviously qualify as proper enzymes or real enzymes. So there's a whole lot of those enzymes. And it's also estimated that those enzymes, the different enzymes that are found in nature, around about 700 chemical reactions are actually controlled by enzymes in prokaryotes, like your bacteria. And then in eukaryotes, like myself and yourself, there's around about 1,000 of those different chemical reactions that the enzymes has to catalyze. All in all, when the enzymes have so much of these chemical reactions to catalyze, they never lose their specificity. The specificity is due to the fact that each and every enzyme has a specific chemical reaction that it must actually catalyze. An enzyme that is responsible for catalyzing a reaction where it involves the breakdown of proteins will not do that to lipids. There will be one that's specific for lipids, like your amylase for starch, your protease for protein. So that's where the specificity comes in. So when these enzymes do all that, they obviously convert your nutrients to energy, and that obviously helps the living organisms to continue with its life processes. In terms of our food science, that is why we have to talk about enzymes, because enzymes are actually applied in food products. There's a whole lot of food products out there that rely on the use of enzymes to actually bring about the food. Make example of your bread baking. You need enzymes from your yeast to obviously break down the starch. You need your enzyme Rene for the coagulation in order to produce your cheese. There's a whole lot of them. But in those instances, there's the good ones whereby it helps us in terms of the processes, but there's also the bad ones that we also need to control. I'll just make an example of polyphenol oxidase, which is a class of what we call oxidoreductases. We'll talk about that later. They catalyze oxidation reduction reactions in the presence of oxygen. They are desirable in food products like your tea. When green tea is being fermented to black tea, we require those enzymes to speed up the process, which is desirable because from green tea to black tea, then you have varieties of teas. Think about green rooibos tea and fermented rooibos tea, green and red rooibos tea. So that is also desirable. However, the very same enzyme, polyphenol oxidase, is the one that brings about browning in plants, in fruits and vegetables, like the browning of your potatoes, your apples, and your lettuce, even some form of seafoods like your I won't say oysters, your shrimps. Your shrimp can also brown due to PPO. So it means that if these enzymes are both desirable and undesirable in terms of food application, it means this should be some form of control in order to ensure that the quality of the food products are actually maintained. So before actually going into the actual enzyme and what it does, let's just first talk about the classification of these enzymes. So these enzymes are actually placed in different groups based on the reaction they catalyze. So before we give them names, individual names, we have to first group them based on what they do, what kind of reaction do they do, and that will be the common reaction that they catalyze. So, and this is actually based on the Enzyme Commission, the International Union of Pure and Applied Chemistry. So they actually do this classification. So they are classified under 6 main big groups, and then there's also subgroups in there. So the first main group, and if you look at some textbooks, you will see that they classify this based on the EC number. So the EC number ranges from EC1 till EC6. Okay, so of the first EC1 category or classification will be what we call the oxidoreductases. You know, the nicest thing about chemistry is that you can break down a word and it's much easier to understand. So oxidoreductases catalyze oxidation reduction reactions, okay? And they're actually referred to as dehydrogenases, oxidases, peroxidases, oxygenases, and reductases. And this is one of those that are very familiar to us, like the polyphenol oxidase that I just mentioned just now falls under these ones. think about the peroxidases when you guys did the blanching of fruits, of vegetables in the frozen vegetable practical. So that enzyme that browns the cauliflower falls under the oxidoreductases. So peroxidases are used to test the effectiveness of blanching in vegetables. And then secondly, the EC2, we have what we call the transferases. The transferases, they catalyze the group transfer reactions, whereby one group is being transferred to another molecule. And then this mostly needs the presence of coenzymes. We'll talk about coenzymes later. And then of the example, it includes the kinases. And then number 3, we have EC3, the hydrolases. The hydrolases, we are also very familiar with those ones. They catalyze hydrolysis. Remember, hydrolysis is the opposite of condensation and polymerization, because with hydrolysis is when the bond has been broken in the presence of water being added there. So these are the special groups of transferases, like I mentioned, with water serving as an acceptor of the group transferred. So they act on the ester bond like your lipases. So your lipase, when you break down the triglyceride at the ester bond, they also work on the glycosidic bond that makes up the sugars and the starch. They also break down the peptide bonds, separating the amino acids from each other. So this will be the hydrolases. And then next on, we have the lysases. The lysases are EC number 4. Those ones, they actually catalyze non-oxidative and non-hydrolytic elimination reactions, or lysis of a substrate, actually the breaking down, or by creating a double bond. So the example will be the synthase for synthesis. So they catalyze the addition of reaction in cells. Then we have EC5, which are the isomerases. The isomerases, they catalyze structural change within one molecule. So isomerization reactions. So they catalyze the change from L amino acid to D amino acid. Maybe L glucose to D glucose. Trans fatty acid, trans linoleic acid to cis linoleic acid. So that's what they actually do. And then what happens is the isomerases, they are the simplest enzymes reaction, because they only need one substrate and then they form one product. And then lastly, E6 will be the ligases. The ligases, they catalyze reactions called ligation, which is actually joining two substrates. So substrate 1 and substrate 2, that's ligation. So this is actually the opposite of hydrolysis, because with hydrolysis we are breaking down the protein, we are breaking down the sucrose into fructose and glucose. But when it's ligation, it means you are taking fructose and glucose and joining them again to form sucrose. Okay, so they require an input of chemical potential energy, because remember, this is joining things together, it's not breaking down. So that's why there's a requirement for that chemical potential energy. And they are also referred as synthesases, because they are synthesizing. Synthesizing is making up. So that will be the classification, obviously based on the different categories. So that's the first classification. The second classification has to do with the complexity of the structure, or we can just say the structure. So we have what we call simple monomeric enzymes. So we said an enzyme is a protein that folds up. So when we say simple monomeric enzyme, so this will just be a long protein chain that folds up, and that's it. So a short protein chain or long protein chain, but it's got nothing else attached to it. Okay, so things like your digestive enzymes, your pepsin and your sucrase, those are your simple monomeric enzymes. And as we already know, those are usually found in the stomach, so they are resistant to the effect of acid, because the stomach conditions are highly acidic. And then secondly, we have the monomeric with attached groups. So it's not just now the protein only, is the protein, is the amino acid, it's something else that's joined in there. And usually These are other called coenzymes or cofactors. I'll explain this when we get to the protein structure. It's much easier to understand there. So they have coenzyme cofactors, and those can either be a carbohydrate, a lipid, or a metal. Since we spoke about proteins earlier on, we also spoke about simple proteins and conjugated proteins. So we can actually say that this monomeric with attached groups can follow under the conjugated proteins, because it's got an amino type of structure and other things that are not amino acids. So this is something similar. Then you have multimeric. So multimeric, you actually have more than one protein chain and more than one active site. I'll still talk about the active site. So you have one protein chain and more than one active site, and these ones are usually present in sigmoidal kinetics. And then you also have the multi-enzyme complexes. So what happens here is actually an aggregate of a number of different enzymes. With the multimeric, it's the same enzymes, but it just contains more than one chain. But when we talk about the multimeric complexes, you actually have an aggregate of number of different enzymes that do different jobs within that whole system. Okay, so all enzymes in that complex catalyze a series of related reactions, but they are different. So an example will be the fatty acid synthase complex. Okay, and then the third classification would be based on their specificity. Okay, so when we talk about enzyme, when they're catalyzing a reaction, each and every enzyme will require a substrate to work with. Okay, so but then depending on the type of enzyme and the type of substrate, we can actually also denote the specificity of that enzyme. How specific is that enzyme for that substrate? Okay, so we have what we call absolute specificity, so that this enzyme can only catalyze only one type of reaction or one type of substrate. That's it. It cannot catalyze related reactions, only one. So that's called highly specific or absolute specificity. And then we also have what we call group specificity, whereby the enzyme catalyzes reactions of molecules with specific functional groups only. So of the functional groups that you guys still remember that you did in carbohydrates, you have your ketones, you have your aldehydes, you have your methyls. So meaning that if this enzyme is catalyzing a reaction, it will only go for the aldehyde group only. And then we have linkage specificity, that the enzyme can only catalyze a reaction which the molecules contain certain bonds. So just say certain bonds, it will only catalyze those kind of bonds. Maybe if it's ester bonds, it can only catalyze the different ester bonds. If it's glycosidic bond, it means it can catalyze only glycosidic bonds, so meaning that it can do 1:4 1.4 glycosidic bond or 1.6, but it has to be that specific linkage. And then we also have what we call the stereochemical. That's whereby it catalyzes the reaction on optical isomers. Okay, so stereochemical, optical steric isomers. So it can maybe only catalyze the trans or the cis form of a certain molecule or reaction involving those, and that's what it does. Okay, so enzyme classification, you classified, just to recap, you classified based on the reaction, the group reactions that they catalyze, based on the structure, and then based on the specificity as to what chemical reactions they actually catalyze. So from there then, we've been talking a lot about the enzyme, this and that, but it's much easier for us to understand the enzyme and how it functions by first looking at the enzyme structure. Like we had already mentioned that enzymes are mostly globular proteins with what we call an active site. So the active site is where the substrate that has to be catalyzed will attach. So it attaches at the active site. So the protein structure is actually divided into different segments. Okay, so segment 1, you have what we call the protein part. The protein part is obviously the amino acids that are joined together by peptide bond, and it becomes a polypeptide that folds on its own depending on the native conformation. So that's the protein part is also called the apoenzyme. On that apoenzyme, you actually have a space known as the active site. So the active site is where the chemical reaction takes place, where the substrate is going to join. And then, depending on the type of enzyme, remember it can either be a monomeric enzyme. So a monomeric enzyme will be just an apoenzyme with an active site and the protein part. However, when it is an enzyme that is joined with something else, it then is joined or attached to what we call a cofactor. If you just go back here, we mentioned that simple monomeric, so that will be the apoenzymes, and then monomeric with attached groups. So this will be the attachment. So the attachment is called the cofactor. So that cofactor can either be a carbohydrate, it can be a lipid, it can be a metal, it can be anything else that is specific for that enzyme. So the cofactor usually is connected or is situated on the active site. And when the cofactor, when the protein or the enzyme contains a cofactor, it becomes what we call a holoenzyme or a complete enzyme, a functional enzyme. So depending on whether that cofactor is actually tightly or loosely bound on the active site, you can either call it a coenzyme or a prosthetic group. So it's called coenzyme when it's loosely attached to the active site. And then it's called a prosthetic group when it's firmly attached. Because, I mean, think about somebody with a prosthetic arm or a prosthetic leg. It cannot be just loosely attached, or otherwise it's going to fall off. So use that analogy to explain between the coenzyme and prosthetic group. So as we already mentioned, that we also have the monomeric enzyme, which just contains the protein part. It does not mean that it does not function. It does function without a cofactor. However, one enzyme that is supposed to have a cofactor, and when it does not have the cofactor, when the cofactor is removed, then that enzyme cannot function. And removal of the cofactor is one of the ways that we actually use to control enzyme activity, because we know it's an integral part of the enzyme. So by removing it, then we are able to control or eliminate that enzyme. But we'll talk about that later. Okay, so after knowing about the structure of the enzyme, then we have to talk about the enzyme action. So what does the enzyme do? So we mentioned that the enzyme contains an active site and then the substrate that is suitable for that enzyme according to its specificity, whether it's absolute or group or linkage, whatever it is. So the active site will be a place where the substrate actually interacts with the enzyme. So let's look at this chemical reaction taking place here. First of all, we have the substrate. The substrate is sucrose. Sucrose, as we already know, it's a sugar, it's a disaccharide. It's made up of glucose and fructose, which are then bonded together with the glycosidic bond, 1.4 glycosidic bond. You still remember that from carbohydrates? So then what happens is, in this reaction, we have sucrose and we want to produce simple glucose and fructose from there. So we want to break down. So this type of reaction will be what we call catabolism, because we are breaking down. So what happens is the sucrose approaches the enzyme. When it approaches the enzyme, then they form what we call the enzyme substrate complex. Okay? And then by forming the substrate complex, it means that the substrate binds to the enzyme on the active site, not anywhere around, but on the active site. So when the substrate is bonded to the active site, what happens is that then the enzymes start functioning, starting to shake the bonds, wanting to break them, so that we end up with what? With what we call the products. So then from there, you also form what we call the enzyme product complex, because now the product is formed but it's not yet separated. So the water is actually added in there, because remember, water in terms of forming that sucrose was actually released. So this is hydrolysis, catabolism hydrolysis, whereby water is used. Okay, so when the water is used here, it's added. So then what happens is that you end up forming the enzyme product substrate, whereby both the products are still attached to the what? To the enzyme, sucrase. Okay, and then they are released after that. So what happens is that the enzyme would have shook the bonds and stressed the bonds until it breaks. If we wanted to accomplish this reaction with nothing, it will probably take 100 million years if you just leave sugar there on its own, sucrose, and hoping it's going to break down to fructose and glucose. But if you want to speed it up, you can use heat, maybe with acid or alkali, but the heat temperatures will be quite high and the time will be a bit longer. However, by using an enzyme, you are then using a biocatalyst that's going to speed up the reaction. And one thing for sure, we can already see in this whole process, the only thing that is consumed is the substrate. The substrate changes and becomes something else. So we end up with glucose and fructose being released as products. However, look at the enzyme. The enzyme is still in the same shape. So it means that if we had another molecule of fructose, of sucrose, we can actually reuse the enzyme. So the enzyme is a biocatalyst that gets used over and over again. It never gets used up, so it can be used again in the reaction. However, the only thing that's actually the limiting factor will be the substrate. If all the substrate is consumed, then we don't have anything or no job for the actual enzyme. And then in terms of the enzyme action, so we already mentioned that, okay, the substrate will definitely have to fit on the actual enzyme for the reaction to take place. So there's two ways of explaining this. We have what we call the lock and key theory versus the induced fit. So the lock and key theory is one of the oldest theory that has been used. And in this lock and key theory, what happens is that it is assumed and known, or it has been proven that in the lock and key theory, the substrate has the exact shape of the active site of the enzyme. So that's what the lock and key theory or hypothesis says. The substrate has the perfect shape to the active site of the enzyme. And in that way, that is why when the substrate gets in there, it just fits in automatically and then the reaction takes place. However, in terms of the induced fit model or theory, what happens is they have hypothesized that the active site does not have the exact shape as the substrate. It's that when the substrate approaches the enzyme would know which substrate obviously it has to react with, because it's inherent in terms of the reaction to take place. But what happens is with the induced fit is that as the substrate gets in contact with the enzyme itself changes the shape to accommodate the substrate. But it can only change the shape for a certain substrate. Amylase will not decide today, you know what, let me change my shape and allow fats to come in. No, it doesn't work like that. So the substrate being fat will still have its own shape, but it will only be catalyzed by your lipases. So as the substrate with a certain shape approaches an enzyme that does not have a perfect shape, what happens is when the enzyme actually or the substrate sits on the active site, the enzyme itself changes shape slightly to actually accommodate the substrate, and in that way the chemical reaction takes place. So in both theories, what happens is the substrate does eventually connect there and then the chemical reaction takes place. But with the induced fit, it tells you about how the shape of the active site actually modifies in order to be able to accommodate the actual substrate. But all in all, what happens is that for the reaction to actually take place, that active site has a specific amino acids, sequence of amino acids. And if that sequence is changed, then the enzyme becomes denatured, or it's mutant and it cannot do what it's supposed to. Okay, so when the substrate actually joins into the enzyme, what happens is that that active site forms a microenvironment for the substrate. So it creates a perfect condition for the reaction to take place. So after binding in there, the substrate becomes distorted, so no, not extortion, sorry, becomes distorted, and then the structure of the substrate then changes, and by then it's actually forcing it into what we call the transition state. So it gets in there. Think about when you are being hugged by a big person. Have you ever been hugged by someone who's big? And when they hug you, it's like you have disappeared in there. That is more or less something analogy-wise. Please do not give this to me in a test. So what happens is that when the substrate fits into the active site, the enzyme accommodates it and distorts the structure of the substrate. And by that distortion is actually forcing it into a transition state by stressing the bonds to break or pushing the bonds together. I'll also make an example of, I don't know if you guys have seen the movie The Anaconda. Have you seen how the anaconda coils somebody and breaks down their bonds in order for them to swallow them? Because they only swallow them when the bonds have been broken, you know, the bones and stuff like that. So same applies to enzyme activity. The enzyme substrate complex, the enzyme then distorts the substrate. By distorting it, it's actually stressing the bonds to either align well for that anabolism to form, or stressing the bonds for catabolism, for the bonds to actually break. And in that way, it takes them into what we call the transition state. But what is this transition state? So the transition state, you know, when you transition, you are in transit to somewhere, okay? The transition state is an unstable arrangement of atoms in which chemical bonds in the process are in the process of being formed or being broken. Okay, so I usually make an example of, think about yourself. Let's say you are at the bottom of the hill and you have a cat that you want to play cards. If you grew up playing with cats or cat box and you want to slide down the slope. So for you to be able to slide, you need to go up the hill. Okay, so you have to push your box or your cat up the hill. On the hill there, once you are on the hill, gravity will obviously do what it does best. So you going up that hill, that's the transition state, the state whereby you are about to slide down. I hope that makes sense. So what happens is the enzyme, in terms of an enzyme catalyzed reactions, are the one that takes the substance into what we call a transition state. And that transition state is just the enzyme actually allow reactants to proceed along pathways that have what we call a lower activation energy, because the activation energy is the one that you require to give a push to get to that top of the hill. That's the activation energy, the push. However, if it's a chemical reaction that's not catalyzed by an enzyme, that energy must come from either heating or rubbing or whatsoever. But there is some form of energy, and usually it's in the form of heat. But with the enzymes, you don't need that. The enzyme will do the distorting and stressing the bonds of the proteins or of the fats without having to apply any heat. So that is why we say the enzyme allow reactants to proceed along pathways that have a lowered activation energy. Okay, so all these chemical reactions obviously are happening at the active site. What can also happen in that active site is that you can get what we call inhibitors and activators. So inhibitors are actually other molecules that would look similar in shape to the substrate. So what they usually do is when they get to the active site, they just get in there, but because they are not specific to that enzyme, the reaction does not take place. But then it's actually blocking the actual substrate. And then you also have what we call activators. Activators what they do is they also bind to the enzyme, and binding to the enzyme, they increase the enzyme activity. So usually molecules that usually bind, they always bind at the active site. Most of the times they would take the active site, and then that's when the actual substrate cannot actually bind in there. Thank you.